The title of my lecture is Nature's Gift to Science. This is not about one journal trying to be polite to another, but it will be on how biological diversity can be and has been used to advance scientific research. I like to think that the fourth winner of the Nobel Prize this year is Caenorhabditis elegans. And of course, I think it deserves most of the honor, although of course it will not share in the monetary reward. I intend to tell you a little about the early work on the nematode, to try to put it into an intellectual perspective, bridging as it did, both in time and conception, contemporary biology as practiced today, and the biology revolution that was initiated some 50 years ago with the discovery of the double helical structure of DNA. My colleagues who follow will tell you more about the worm and also about their incisive researches on it. To begin with, I can do no better than to quote the opening sentence of my paper in 1974. The paper was unhesitatingly entitled The Genetics of Caenorhabditis elegans. And the opening sentence reads, how genes might specify the complex structures found in higher organisms is a major unsolved problem of biology. It still remains true today. Indeed, 20 years before that, we were writing in a similar vein such sentences as the central question in biology was how the one-dimensional sequence in DNA specifies the one-dimensional sequence of amino acids in protein. In the early 60s, that question was laid to rest with the discovery of the genetic code, which actually told us how to read the one language, the language of the gene, into the language of function, the language of proteins. Of course, once that had been solved, in principle, though much detail remained to be worked out, attention turned to the problems of the more complex structures in biological systems. And indeed, the same question was asked in that paper and could be said, how does the genotype for complex structures map onto the phenotype that we can study? Now, I will just go back for a moment to some of the more classical work on molecular biology. It became clear at one stage that if one wished to try to find out how one-dimensional sequence of bases specifies the one-dimensional sequence of proteins, it would be very easy. All you have to do is get a gene and sequence it, and get the protein and sequence it, and you can just translate one into the other. Of course, we didn't have those tools at the time. We had tools to sequence proteins. They were still very primitive, but at least we could tackle the protein chemically. But there was no way to tackle the gene chemically. All we could do was analyze the gene through the phenotype. through mutations, following the standard path, first actually stated by Mendel, namely the only way to say that there is a gene existing in an organism is to find a mutant allele of it. So Mendel could not say there was a gene for tallness until he discovered plants suffering from a lack of tallness, namely dwarf mutants. And classical genetics, through the phenotype of mutants, offers a view of the gene which you can do by making observations on how the phenotypes are displayed in crosses of two organisms. But to dissect the structure of the gene required something rather special. And effectively, that special case was provided by bacteriophages. In bacteriophages, with the discovery of recombination, one could, because of their rapid multiplication, because that their very large numbers could be handled very conveniently, it was possible, and indeed that was Benzer's fundamental contribution, it was possible to make a dissection of the fine structure of a gene and show, effectively destroy the classical concept of a gene that was both a unit of recombination, mutation, and function, and show that it was composed of many, many mutant sites. And the important thing was that the resolution of recombination could be carried down to a level that corresponded to the distance between adjacent bases on the physical structure of DNA, so that we could look, so to speak, at the sequence indirectly by crossing two bacteriophages and measuring how frequently we got recombinants from it. And of course, further work done with bacteriophages, with bacteria as model systems, allowed us first to come to understand the genetics of regulation by the work of Jacob and Monod. And so it was very natural for people in this field of molecular genetics to say, is there a parallel way of tackling complex structures in organisms? The path was very clear. What one wants is an organism that one could perform genetics very rapidly, that had a rapid growth rate, that was finite. Because not only does one need the capacity to do genetics, but one needs the capacity to analyze the phenotype. Because the rule at that time was obtain mutants and study them as deeply as you can. The tool I chose was electron microscopy, because I was interested in the nervous system. And what I had thought was important to do was to study exactly what could be summarized in the form of the wiring diagram. And again, in the paper, we planned not only not to go directly from gene to behavior, but to separate the problem in two parts. Namely, one, a developmental problem, which is how do genes build nervous systems? And two, the physiological problem, which is how do nervous systems or brains work to generate behavior? And so we started by doing the serial section reconstruction of C. elegans, and roughly at the same time trying to search for mutants. The paper referred to summarized the work on genetics, and of course, at that time, in the early 70s, the chances of obtaining a molecular analysis, even in an organism as simple as C. elegans, were remote. If one asked, how would one find out what the products of these genes were, that seemed extremely difficult. We started to do molecular biology there by studying muscle, simply because we could get hold of large amounts of proteins. And the idea was, well, perhaps myosin is the beta-galactosidase of C. elegans, and so we'll study structural mutants there, and then one day, if we're lucky, we'll start to study regulatory mutants. And of course, I doubt whether any of us would be standing here today were it not for the technology invented in the mid-1970s, which allowed us to clone DNA and to sequence it, to characterize it directly. And I think that that then opened up the whole new field, a new way of doing genetics. Indeed, although we had proudly said that our work in model organisms would illuminate the molecular biology of higher organisms, including ourselves, it was a very remarkable fact that when we came to characterize the gene products of the genes that had caused morphological changes, we found they'd already been discovered before in human beings. And so, as you will hear, one of the important genes turns out to specify something called RAS, which of course was in a database and had already been described in human beings. So it could be said that the work done by these new techniques on humans and mammalian organisms illuminated enormously the work done for us in model organisms like C. elegans and Drosophila. Well, there are many other examples of model organisms being used when our technology is limiting. I think the most important thing that I know about was the ability to study in detail with rather primitive tools the nerve impulse, simply because they happen to exist in squids very, very large axons. And it was able to perform on these very, very large axons, almost a millimeter in diameter, experiments which could not be performed in any other way on single nerve fibers at the time. And so this interplay of nature and science has been very important in the development of our subject. And so when time had moved on and one had become now in possession of new ways of doing genetics, one began to wonder whether there were other fortunate models. And I will just recount one, which, again, in much the same vein, came with the whole business of sequencing genomes in complex organisms. I happened in the end of the 1960s spend a lot of time at Woods Hole, which had a marvelous library in which one could read lots of journals that you can't find anywhere else. And in reading a journal called The American Naturalist, which most people think is a nudist magazine, I came across papers by a man called Heimgartner showing that certain fish had very small genomes indeed. And of course, at the time, we didn't understand the problems of complex organisms at all. And if you asked people, does it make sense that this fish has a genome 1/8 the size of ours? Most people said, yes, that figures. I'm 8 times smarter than a fish. I'm 8 times better looking than a fish. And it's got the number of genes it deserves. Now, of course, a fish is a vertebrate, just like us. It has many of the constituent parts. It has an immune system that's very similar to ours. It has a neuropeptide system that's very similar to ours. And so, was it possible, one asked, that here, embodied in this very compact genome, there is a quick way or a direct way of obtaining basic knowledge about vertebrate genes, which can be applied to our studies of other genomes. Because at the end of the day, we are going to have to ask and answer the question of what is the model organism for us human beings? Well, it so happened that after many, many years, we were able to characterize the genome of the fish, which will be added to the worm as another model organism. And it's quite right, because someone pointed out to me that all my work had been done on organisms that eat each other, that I had started with E. coli, and then I used worms which I fed on E. coli, and then I had started on the Japanese pufferfish, which eats worms, probably. And so I suppose the next thing I have to do is the Japanese genome. But I think it is well to pause now and ask a most interesting question, which is, what are the problems now remaining with this great surge of new approaches to the complex biology? In fact, our task for the future could be said that we need to understand biological complexity. And I think if I can, I will single out 2 areas where I think we need to sit back and reflect on the tasks that lie ahead. And perhaps the best of these could be, the best way to think about these is in the form of a program, or probably better still, a manifesto. As everybody knows, today we are all drowning in a sea of data. And of course, we're all starving for knowledge. We have unprecedented amount of basic facts of biology, basic descriptions pouring out. We need to understand that. And the question is, into what perspective do we place this understanding? I think it's quite interesting to note that many people have forgotten what the basic units of function in a living organism are. They're not the genes. Now, something that goes back much, much earlier than that. They are cells. And in fact, in C. elegans, we asked the questions about cells, not so much about genes, because it is the genes that work in cells that effectively give us the function of the organism. And the perspective must be in terms of the cells of an organism. As the president said when he began, how does this complex structure that is any multicellular organism arise? And of course, our job is first to say how many different cells there are in such an organism. I think we can say that completely for C. elegans, and you will hear later how each cell can be strictly defined, and in fact we are now well on the way in the worm to saying what are the sub-programs, if you like, of the genes that are working there. But for vertebrate organisms like ourselves, we are very far away. If you look at a textbook of histology, there may be 200 different cell types in the body. But how many cell types are there in the brain? How many different cells are there? There could be at least as many as that, and there could be even more. There could be 1,000 cell types. So I think the task then is to continue what we started with C. elegans now and to carry it forward into higher organisms. Because now we don't have to perform crosses to tell us where mutants lie on a gene. That can just be done directly because we can read the genes directly. And so I think that one program in which I think modern knowledge will have to be embedded is a program of making maps of cells and making maps of how cells talk to each other. So, the Cell Map Project, as I call it, of which we don't need a model organism to do that anymore, will be one of the things that will occupy us for the next few decades. But there is something else that I think we need to take into account, and that is that humanity now becomes a subject for biological research. And I say humanity and not the human genome. I was struck by this by attending a meeting the other day, which was on the mouse. The mouse, as you know, is thought to be the model for man. The meeting proposed that we take the inbred strains of mice, cross them together, create a pool of 30,000 mice that were all mixed up. Then we would train a whole lot of people called mouse phenotypers that would go around, examine these mice and diagnose high blood pressure, diabetes, greed, flatulence, all the common ailments of life. And then we would study the genotypes of these. Except we don't have technology that's reasonable enough to do 30,000 genotypes. And I reflected that if one had the technology to look at, let us say, 100,000 genotypes, would one work on mouse? The answer is no, because that experiment of mixing everything up has already been done. We've done it ourselves, outside. And we have trained very expensive human phenotypers called doctors. And they will tell us what is wrong with those people. So if the technology exists, and I think it can exist and will exist, why don't we work directly on humanity? And I think that the idea of looking at ourselves in this way, and if one works with a population where everybody is anonymous, that is the right way to do human biology. So one shouldn't work on cases and controls, one should work on populations. So it may be that in this century, the model organism will only be used to confirm what we find in the real thing. And just as a chemist today will prove that he understands the structure of a compound that he has found by degrading the compound, and he proves it by synthesizing it, so we will prove that we understand the genetic structure of a human disease by synthesizing the same disease in a mouse. Indeed, already this has been done. And I think that that is a prototype in Hirschsprung's disease by Chakrabarty. So I think that if we can move our attention to man, I think that this will be the future. And finally, if you ask, perhaps there is a really interesting twist to this, because maybe man is a model organism for the gods. And maybe if we wish to understand things that are beyond us, like morality, ethics, which are supposed to be the properties of the gods, that maybe we can study them in ourselves, the model organism, mankind. Thank you very much.